Finally, we're thrilled to welcome Dr. Daniel Geschwind. Dr. Geschwind is the Gordon and Virginia Distinguished Professor of Neurology, Psychiatry, and Human Genetics. He's a Senior Associate Dean and Associate Vice Chancellor for Precision Health at UCLA. Pioneering work in the Geschwind Lab integrates population genetics, functional genomics, and bioinformatics with basic and clinical neuroscience to advance our understanding of neurologic and psychiatric disease, pathogenesis, and to accelerate treatment development. Dr. Geschwind, we're incredibly honored you're here, and the floor is yours.
Okay, so I'm going to talk about, just to give a little background, it will be obvious, my lab is not an expert in chromatin biology at all. We're interested in understanding how human genetic variation actually impacts the brain and brain diseases. So we're trying to use these approaches to connect genetic variation that increases susceptibility for human brain diseases, mostly neuropsychiatric, to understand their genetic mechanisms, and then of course eventually the kind of cellular molecular mechanisms. So I'm going to give a kind of high, at some level maybe, I'll go into some details in some areas, but mostly go over what we've been doing. And I wanted to acknowledge key people in my lab. This is a newer picture on the right that doesn't include an essential person, Hae-Jung Won, who really started doing Hi-C in my lab and brought Jason Stein and Luis de la Torre Viedo, who worked with her initially and also both in Hi-C a little bit, but more so in other chromatin explorations. It will become clear using ATAC-seg and other data. All the starred people now are gone. Hae-Jung and Jason are both at UNC Chapel Hill in genetics and neuroscience with their own separate labs. Luis is at UCLA. Neil's in a neurology residency. Mike's at UCLA. Vivek's at UCI. And then I'm going to talk about some work that's unpublished that Jenny Yin's been working on, who's a very talented postdoc in the lab. And some of that work has been done in collaboration with Alison Goat at Mount Sinai School of Medicine. So I wanted to put this first in case I run out of time, because I tend to. And of course, we're grateful to our funding sources, and we've been, a lot of what we're doing now is incorporated within the PsychEncode project. My talk's going to be in two pieces. One, I'm going to talk about neuropsychiatric and developmental disorders, and the second, exploring the impact of structural variation. So just to start with the first part, neuropsychiatric diseases have strong genetic components. Their prevalence is high. Their twin kind of family-based heritability and SNP-based heritability is significant and substantial. And the main point I want to make here is that the genetic contributions are complex, and that as the sample sizes have increased, shown on the left and the y-axis in D, schizophrenia being the big one, there one can see it's not quite a monotonic relationship at all, but there's a relationship between the number of samples studied and the number of loci identified. So loci. So anyway, the point being that there's a lot of gene finding going on. It's really highlighting the complexity of these disorders. And this is just showing some work in autism. That's just a summary from a few years ago that shows not only are we identifying common variants through GWAS, but especially in conditions like autism, the earlier, more severe conditions, we're identifying maybe up to 20% of patients who have a large contributory, some people might say causal mutation. And these are comprised of large effect size mutations in a number of genes, none of which really account for more than 1% of the syndrome. You can see in parentheses what was the contribution at the time. So it's a kind of this 20% is even a collection of rare disorders, and we can even put on top of that common variation. So we have a complex picture, but our job is to kind of embrace this complexity because it is what it is, and then to understand mechanisms of disease from this. And I think we have all the tools in hand to begin to do this. So I'm very optimistic. We just have to not put our head in the sand. So advances in genetics and genomics now make it possible to identify genes involved in susceptibility for common neuropsychiatric disorders. The same goes for neurodegenerative disorders, et cetera. However, they highlight extreme heterogeneity, even in kind of rare, conditions with rare forms like autism. Individual genes don't act alone. They act within pathways and networks, and they're obviously genetic sequence effects are modified by these other factors. And so our approach the whole way along has been to scratch our head and try to use integrative systems biology approaches to identify pathways and networks and translate genetic findings into mechanisms. And, and we have all kinds of interesting questions around this. Will we, despite the heterogeneity at the etiologic side, are there areas of convergence like there are in most disorders? And the answer is likely yes, but we don't know exactly where and when this will occur yet. But this is one of the main things we've been working on. So I'm going to start with just showing you some of that so you can see how that informs what we're doing with epigenetics. So this first paper came out in 2013, and it was a study that was based on scant genetic information at the time. In 2012, a series of exome sequencing papers came out in autism that I think combined had fewer than 1,000 probands. And we took those genes, which were, that were hit at least once with protein disrupting de novo mutations, and asked if we created developmental brain networks in normal brain development and looked at the trajectory of the transcriptome, could we identify, were these genes randomly distributed, or were they distributed in very specific pathways? And it turns out they were very non-random, especially if we compared it to intellectual disability. And this is just showing one module called M3 that was enriched in chromatin modifying genes. In fact, the two major developmental modules expressed at the peak of cortical neurogenesis, which is between 13 and 20 weeks gestation, all were either enriched in transcription factors or chromatin modifiers, probably involved in key aspects of pattern formation and neurogenesis and fate, cell fate, et cetera, and morphology and differentiation. And so this was pretty remarkable, especially when we compared it to intellectual disability, which showed remarkably less specificity at the time, given the same sample size. Now I could say that the genes that affect intellectual disability, because there are many more that have been identified through exome sequencing, actually have some overlap with these pathways as well, but it's not complete. So what we had at the time was that if we compared rare de novo variants in autism to those that were differentially expressed in postmortem brain, to candidate genes and GWAS hits that were subthreshold, compared that to intellectual disability, and then asked about molecular networks, in other words, co-expression networks, genes that were highly co-expressed during brain development, we essentially saw kind of two major patterns. And it's just shown here on the right that you would have, and figure birth is kind of occurring right in the middle of this graph showing transcriptional regulation and synaptic, so birth would be there, that you have these genes that are de novo, large effect size mutations as shown in the M3 at the left are expressed early at the peak of neurogenesis, and they go down to the point where they're very lowly expressed in postnatal brain as a group. And then synaptic genes and signaling genes kind of go up. They're expressed during mid-gestation, but they continue to go up as synaptic development proceeds over birth. And so what we have there is we saw that autism genes converge to disrupt what we call neural development and cortical connectivity. Why do we say that? Because at the time, there was a enrichment of risk genes in these superficial cortical layers, layers 4 and 2, 3, which are the layers that connect different cortical regions to each other and connect the two hemispheres. Some of the genes do impact layer 5 and 6, which project down out of the cortex. They definitely do, but it's less of an enrichment than for the superficial genes. What was interesting about that from a molecular standpoint is that that also overlaps with imaging findings that showed disruption in long-range connectivity. But this kind of beginning look at this, and when you look at the network on the left, we don't understand what most of these genes do during brain development. Many of them have been identified in cancer or in other cell types and have some known function, but it really led us to this following thought. And let me just emphasize again that the genes, the de novo mutations in red are occurring in these transcriptional regulators, and the synaptic development genes are more likely to be those impacted by lower effect size variants. So it led us to kind of thinking in this way. Again, if you think from an evolutionary and population genetics standpoint, de novo mutations are new and they have very large effects, so they haven't been removed by natural selection, whereas things that are shared in the population are ancient and common. Those are the blue things. And if we kind of overlap those onto the two main functions that we saw disrupted, we see that transcriptional regulation are the de novos, and synaptic development function are more enriched in the common. And if we kind of now ask, where does this fit in terms of what's known about transcriptional regulation and transcriptional regulatory networks? There's a sense of a kind of tiered networks that are either 3 or 4 tiers high, with the top tier regulators being transcription factors and major, whose effects then are on the expression or splicing of other genes. So the top tier regulators are those kind of regulatory genes that have widespread effects, and so it fits in a way. This genetic architecture that we're seeing parallels what you might predict from a molecular regulatory architecture, in that large effect size mutations are more likely, not restricted to, but more likely to be in genes that have large scale effects that are at the top tier, because those effects kind of percolate through the network. But this really highlighted to us early in 2012 and 13 the need for having a refined understanding of regulatory networks in brain development if we really wanted to understand what these genes were doing. Now, I cite a review here. There's quite a bit of work going on in this, in this area. It's not as if there was nothing known, but really, if we compare it to other lineage studies, like in hematopoietic system, we're really far from that. And we really needed to move into that area. And another thing that really kind of hit that we recognized at the time is that most of the GWAS hits were sitting in non-coding areas. And so if we wanted to understand how they were acting and what genes they were acting, we had to better annotate those non-coding areas with respect to brain, especially because biology is driven by gene regulation that occurs at the cell type and tissue level. The notion that it's not just what's going to be open, what's going to be active, what transcription factors are expressed, all of those things go into how a gene is going to be turned on. And a lot of that is cell type, not tissue specific or stage specific. And then we just heard, I got on at the end because I had something else going on and I apologize, but about activity dependence. And of course, that's a whole nother dimension that's extraordinarily important for the nervous system. So that's like, let's call that the 5th dimension. And so, recognizing that, we realized there was really very, very little known about, especially about developing human brain. And this is summarized in this title of this wonderful movie, if you like science fiction, on William Gibson, that my wife and I helped produce a long time ago when our 20-year-old was 6 months old, because he's in the movie. But this title, No Maps for These Territories, which speaks about the future, really resonated with me as I was thinking in 2013 and 14 about kind of what we needed to do. And I wasn't sure what tools there were until Haejung really came to the lab. Another set of, there were a bunch of papers popping out too identifying pathways that were affected in psychiatric disorders, and implicating histone and chromatin remodeling as well. So there was, this other kind of, a lot of literature pointing to this. So, and we had started a study in 2011 that took a long time with, they all do when we're studying human brain, a histone acetylone-wide study of autism, taking 2 cortical regions and cerebellum with Sham Prabhakar at GIS, looking at H3K27 acetylation patterns. And again, we showed, there was a shared kind of chromatin enhancer pattern disrupted in autism that actually corresponded quite well to the transcriptome dysregulation that we had identified before. So again, just a lot of evidence telling us, boy, we need to look in this area and we need to make better maps. So what Hae-Jung started with was to map chromosome conformation using Hi-C during human brain development and understand its relationship to gene regulation. And we started this soon after the first Hi-C papers had come out in 2013 from Job Decker, Bing Ren, Lieberman, and then Brode. And then the idea was to leverage Lieberman and Aiden, leverage the chromosome conformation data to annotate non-coding regulatory elements and common risk variants for schizophrenia and other disorders. And this, again, was led by Hae-Jung Won, and some of the other work I'll discuss was also with Jason Stein and Luis de la Torre Ubieda. So again, we're generating a reference map of gene regulation in fetal human cortex. And the idea was, we started with a very simplistic notion, but we wanted to have some known biology as a guide for us. So what's shown there on the right are the laminations. It's just a DAPI stain of the cortex. And we have the germinal zones on the bottom where neurons are being born. Then they migrate up into the cortical plate through the intermediate zone and subplate. And that area, cortical plate, expands to become the adult cerebral cortex. Or, certainly all those neurons are born before 26 weeks in gestation, again, with a peak kind of between 13 and 18 weeks. So we wanted to know what genes are expressed, what regions of the genome are active, what genes they regulate, and how do they regulate them. And then I cite some of the papers that have started to integrate these data below. But I'm going to start with the Hi-C data. So this is what Hae-Jung did. I'm not going to go over this in detail. This was, again, done around 2014-15. You can see we used a pipeline, kind of what I would call a standard pipeline at that time. And Hae-Jung called loops by fitting a Weibull distribution, which looked appropriate at the time. So the first thing that Hae-Jung did was first, we looked at the other data that was out there. There weren't that many papers yet in other cell lines, papers from Bing Ren and from Job Decker's lab, and we had kind of, we were able to identify, compartments and TADs, and there was a high overlap between our data, our TAD calling and theirs, et cetera. So that made us feel that we had decent data, about a 0.8 correlation or so. What we're really interested in is asking, are these Hi-C loops that we're defining functional? Because the TADs were largely, not 100%, largely preserved over cells. So how do you get cell type gene regulation that's specific? And so we reason that's going to be in the looping, which is bringing enhancers and promoters into contact. And so these maps were made at a 10 kb resolution from a set of n equals 3 for each germinal zone and cortical plate region. And so then Hae-Jung with Irina Buonagu had looked at the relationship between eRNA and mRNA, and we could see that there was a strong correspondence between eRNA, mRNA correlations and loops. We did not have fetal brain EQTL, it was adult brain, but we still asked, are genes that are physically interacting here with Hi-C at the loop level with EQTL, are they significantly associated with EQTL-associated transcripts? The answer was yes. and also used DHS openness promoter correlation and showed a kind of very similar thing. And I'm just going to give a very, very quick example of one of these just to show you what I mean. So the idea is that if a high C interaction between a variant, let's call it, the T and C sitting in the middle, is actually a regulatory interaction, that it should be non-randomly associated with expression quantitative trait loci. In other words, with the gene expression. So, is T associated with gene B, A, or C? And so what we were able to do is basically ask at the overlap. We had this EQTL data set from Swami et al., which was a underpowered but nice data set from human brain with a few, about 150 samples, maybe 120. And so it's of course an incomplete map, and so is our high C map, by the way, because we don't have a huge number of samples. But we could see that if we look both at the germinal zone high C libraries and the cortical plate libraries, that we had very high overlap, quite significant overlap between, the EQTL. It's about a 30-something% overlap between the high C peaks and the EQTL identified. This is highly significant. So it gave us some evidence that what we were looking at were these loops were likely to be en masse regulatory. And so, of course, then we went to ask, can we use this to annotate GWAS signals? And Hei-Jung chose schizophrenia because there had been this beautiful paper in 2014 from PGC that was a turning point in the field by identifying 108 schizophrenia-associated genetic loci. But in only 10 instances was the association signal really clearly attributable to a change within a known gene. And the way that things were done was as follows. You basically look along the genome and you assign SNPs, the index SNPs identified in a GWAS, to the nearest gene. And okay, that, or you could take all genes within the linkage disequilibrium block, the LD block around that SNP, and say, okay, these are the candidate genes. But of course, in some blocks, there might be dozens of genes. So there's still quite a bit of work to be done. And you could do overlap with EQTL, which is a step better, but that wasn't done in this case in that 2014 paper. And there frankly wasn't much EQTL data in brain yet to speak of. And we decided, boy, if these loops are defining functional enhancer promoter interactions, then we could actually assign genes to SNPs to genes using the loops. And another reason why we thought this would be a good idea is because in looking at the known EQTL data, most of which was out of brain, we could see that at least half of EQTL were quite distal. In other words, they were not acting on the closest gene. So it gave us a really substantial reason to think that this would be a better way to go. So we saw, based on the high C data, that only half of the GWAS loci could be mapped to the closest gene using this. And many are acting more than several 100,000 base pairs away, 95% within the same tag, but still that could be hundreds of thousands of base pairs away. This is an example of one locus. The credible SNP is identified there, the GWAS locus, and its credible SNP. We used a mapping technique called caviar, bioinformatic technique. And so there's the GWAS locus with the LD block in pink. Then below it, we have the TADs from the GZ and CP labeled in different colors. And then the negative log 10 p-value of the high C interactions, one there, and a very, very large one there, especially present in cortical plate, which is post-mitotic neurons. And so we look here, we see these SNPs, and we see that what it's interacting with is something entirely outside of the LD block and appears to be this acetylcholine receptor promoter. So we identified by assigning this to the right gene, we were actually able to implicate new pathways, including cortical neurogenesis and acetylcholine receptor activity. There were numerous cholinergic receptors that were identified in this way. And we were also able to validate that quite a few of the SNPs, about half of them were actually interacting with the closest gene in some cases, but you couldn't a priori know that. This is another locus that's just far outside of the LD block and just showing what that looks like. So, you know, again, we saw numerous cases like this. And so what we were able to do is now use CRISPR in primary neural progenitor lines to take out the region surrounding these loci. I'm going to show a different locus that's FOXG1. It's another gene involved in proliferation like SOX2 is, but it was just almost 700 kb away. So it was just so interesting. There's the SNP in between PRKD1 and this non-coding RNA, and there's FOXG1, and one could see that this prediction from the Hi-C map would be that the region that that SNP is contained within, which is a 10 kb region again, let me be clear about that, interacts with the FOXG1, which is hundreds of thousands of base pairs away. And so we used, made a few different snippers with CRISPR and showed that we were able to, that it had a specific effect on the FOXG1 and didn't have much of an effect on the closest gene. And we did this for a number of genes, used some other methods to validate some of these as well. So this, you know, and this is a kind of a nice form of biological validation. Subsequent to that, we've kind of incorporated in the same, using the same model, the germinal zone and cortical plate, with ATAC-seg maps to look directly at chromatin accessibility, which has the advantage that it's also kind of a smaller region. Our average ATAC-seg is about 1,100 base pairs. And we reasoned that if we could take open chromatin that's in a promoter and look within a TAD and find the most correlated in terms of read pileup open chromatin regions, those would likely, you know, be the likely enhancers. And indeed, that's what we saw by overlapping this type of data with Hi-C. We think we got the highest confidence kind of regulatory interactions. So we use this to define a higher resolution map of non-coding regulatory elements active during neurogenesis. And we studied, you know, we did a bunch of studies with it, including reannotating GWAS. And I'm not going to go into the details here. It's published, but I just wanted to make the point that these data need to be, you know, are optimally used when they're integrated with other data. And more recently, defining EQTL in fetal, you know, we finally, after years, made an EQTL in fetal brain. It took us years to collect enough brains. We had 220 brains, and this was led, again, by Jason Stein when he was in the lab, and Luis de la Torre Vieta, but really taken to completion by Rebecca Walker, a graduate student in the lab who's now finished. But you know, the idea was we had, you know, over 200 fetal brains, and so that was a reasonable size. We identified about 6,500 E genes, and we were able to identify splice QTLs as well. And using single cell sequencing, kind of deconvolute some of this to a first approximation to cell type specific patterns. And so we integrated this with looking and asking, you know, where, you know, using LD score regression, you know, where does the heritability for various, you know, disorders sit? We use this for a transcription-wide association study, and we conducted co-expression network analysis to identify modules of co-expressed genes that might be enriched in various disease variants. But one of the points I want to make is, again, just like before, if we compared a similar size data set from adult cerebral cortex, we can see that you know, we're identifying a similar number of E genes, but the overlap is not even 50% in a similar size study. Again, the point being that one needs tissue and stage specific maps here. So one of the things that Rebecca also looked at, like we had before, is asking if we look at the presumed EQTL sitting within enhancers, acting to increase or decrease transcription. We asked about their overlap with ATAC-seg correlations and Hi-C loops. And so if we take all EQTLs, both distal and proximal, distal being further than 10 kb away from the EQTL peak, we could see that a large percentage, more than half, are supported by the ATAC-seg data. In other words, the correlation of enhancers and promoters. Now, for high C, because we only had 10 kb resolution, we could see that it was lower, but it was still quite significant. About 25% of the distal EQTLs overlapped with a high C loop. Now, why is that? It could be that obviously we've only done a total of 12 high C experiments from 3 different brains. So we probably need a much larger and kind of deeper sequenced cohorts to have a more complete high C looping map. But in any case, I'm happy to discuss that. So just to conclude this, and then do I have 5 minutes left to just show a few ARIMA things? Is that okay?
Sure, definitely.
Yeah, so I just wanted to show you kind of where we had been going, and I just summarized that here. That we're able to validate these data, that they're really, really powerful, we think, for connecting loci to genes, enhancers to genes, etc. And we think that integrating with other data may improve the predictive ability. And this is just showing several of the papers where we've made, I'll be honest, relatively minor contributions by basically integrating the high C and ATAC-seg data, these functional genomic data, with GWAS data to actually do a better job of annotating actually what are the likely candidate targets of these GWAS loci. And this, I think, was culminated in our PsycEncode project papers, both with Sham Akbarian and then the Wang et al. With Mark Gerstein, where we integrated adult high C data with other data and really showed how it can increase the power of machine learning, deep learning methods to kind of identify new candidate genes. And all of this kind of culminated in Hei-Jung. She had started this when she was in my lab, but it was her idea and definitely not my idea. This is essentially what we were doing. We were using these tools to assign these functional genomics data to assign SNPs to genes. And so she attached it to Magma, which we had been kind of doing in the lab, but not in a formal way, to create a tool that now allows you, instead of having the genes assigned to the closest gene or the LD block, to actually use these functional genomic data to improve the prediction of risk genes. So I urge you to go take a look at that if you're interested in this. The rest of the work, which has been going on for a couple of years now, is work from Jenny Yin, who came to the lab, and she's doing she's really taken over where Hei-Jung left off and is exploring the impact of structural variation, as well as looking in postmortem autism brain. She has multiple projects, but I just want to discuss the structural variation stuff because it's a kind of another spin on this. Here's the idea. We have a disorder like 15q duplication, which causes about 0.6% of autism. It's highly penetrant for autism. And there's a big region with multiple breakpoints that is duplicated in this syndrome. And one of our notions was, we always think of these CNVs as just affecting the genes within the region, but how about the distal effects? How about effects on gene expression that are resulting from epigenetic changes in TAD boundaries or looping or compartments, et cetera. And so when she started this, rather than just jumping on our ongoing high C, which was working, but quite expensive and needed a lot of tissue, she compared these 3 methods, and she's presented this before, so I'm not going to go into gory detail, but leave me to say that we're able to use the ARIMA high C, probably because of the cocktail approach and some other things, to get really good high C maps from almost a tenth of the number of cells. So it's definitely been an improvement for us. This is data that Jenny's presented before, but I just want to, one point here was that the easy high C in our hands did not work very well. That could be because those are our hands, but compared to the ARIMA high C and our regular in situ high C, you know, the original high C from Lieberman. But you know, we're getting quite good maps. I'm not going to go through all the technical details, but I'm happy to in questions. But one of the points here is that we did this in sorted neurons that were done, given to, where Jenny sorted them with new N positive and new N negative using a protocol that Chamakbarian had given us, and that really works quite well. And so you can have the new N positive on the left, and the new N negative on the right, the controls on top, the 15q duplication on the bottom, and we can see that within cell there's a very high correlation between the high C matrices. I think it's at 100K resolution. Jenny can correct me, but some, you know, it's not a, this is 100K, I think, or something like that. And the same, you know, and you can see again within cell type better than between the cell types, which is kind of what we expected. But good correspondence actually between the maps at this resolution, which is what we expected. And essentially, Jenny went through looking at many methods, and she could discuss her choice. But she did an A/B compartment analysis, a TAD analysis, and of course the looping analysis, just like Haejung had done before. But we're using slightly different methods now. There are a lot more methods on the market, right? And this is one of the major findings to date, is that if we look at the A/B compartments, it's not just in this brain region. We actually can look in other brain regions and see this as well. That within the new N positive cells, we see a big compartment switch inside and around the duplication, but we don't see that in new N negative cells. So this compartment switch is cell type specific. And Jenny is now working on getting deeper libraries and looking at looping and TAD boundaries and all that. I'm not gonna, because of time, I didn't want to go into that, but just show you the main point here was, looks like there is some cell type specificity to this compartment change. And she's done this in I think it's 6 cases in control. So this is not just one case. A second structural variation that Jenny is working on is this famous chromosome 17 inversion that's about 900 kb that leads to a tau haplotype, H1 and H2 it's called. It has a different frequency across different populations, but the H1 haplotype is of interest to us and many others because it is a major risk factor for neurodegenerative diseases, including progressive supranuclear palsy, which is a Parkinson's plus syndrome, in which about 96% of the patients have this H1 haplotype. The tau gene is circled there in the middle. There are probably other things involved. And so our hypothesis is that the gross structural differences between haplotypes will confer effects on the regulation of gene expression in this region, as well as the wider transcriptome. And so this is work done with Alison Goat at Mount Sinai, where in her lab, her postdoc has been growing, and these were made by Celeste Karch originally from patients who were homozygous for H1 or H2 haplotypes. And then they're grown into excitatory neurons, astrocytes, or microglia. I'm just going to be talking about the excitatory neuron work right now on the high C chromatin structure. But one of the really interesting things that Jenny kind of solved is that we realized we had to call loops properly. We had to construct reference genomes of the 2 haplotypes separately. Because if you look to the right, I mean, one uses the distribution of contacts with relevance to their genomic distance to kind of call loops. And if you've misspecified the genomic distance and that it actually is much closer or much further, you've changed your denominator. You're using the wrong denominator. I hope that makes sense. So within this inversion, and so we mapped each onto its respective actual true structure, which took quite a while to do. She used 2 TAD boundary calling programs. She can talk about why she chose them, Top Dome and looking at the insulation score. And the top dome are blue, the insulation score is showing the likely TADs in the dark blue below. She did an N of 3 of each, and so the H1s are shown up above and the H2s below, and one can see that there are quite a few conserved TAD boundaries, especially outside the region, but inside the region we can begin to see changes. And in fact, that's shown here, showing both using the insulation score and the top dome. We can see that there's, if we go between the H1 and H2 and ask for, what are the percentage of TAD boundaries that are shifting, we see, it's pretty low, close to 0, and then it pops up, within this region, which is that huge peak, and then both the insulation score and the top dome show the same thing. So that's telling, that's pretty interesting, and telling us that what we're seeing in this locus is not a artifact of the way we're doing the analysis. It's actually happening within this locus. And so then, Jenny's done a lot more work, but I just, again, wanted to summarize one thing here. This is just showing the consistent loops between the that is, those that are seen in more than one of the samples, and there are many consistent loops that are, that are still there, and the loops are shown in the dark kind of purple, but one can see clearly there have been huge shifts in the TAD boundaries here, and so she can look to see if there are haplotype specific loops, and here are a few that have been identified, and she's now characterizing them in much more detail. But again, this just shows how the kind of high-seed method can be used to look at structural variation and its effect, to begin to understand the mechanisms that affect gene regulation. So the rearrangements between the H1 and H2 haplotypes affect both TAD and loop structures, and we think that they affect many genes within the locus by linking the genes with different enhancers or different methylated CG dinucleotides. Some of the areas that are being linked aren't kind of known enhancers, but are CPG rich. So anyway, thanks for your attention. I thanked everybody up front, and I apologize for going a few minutes over. I just wanted to show some of the new stuff that we're doing and some of the mess that we're in. So anyway, thanks.
Thanks a lot, Dan. Maybe it's time for one or two questions here regarding the first part of the talk, and the utility of integrating chromatin interaction data like Hi-C with other features like open chromatin for linking disease-associated variant regions to target genes. You showed something there about the CRISPR-based validation, and one of the things that commonly I think comes up when I also speak about the utility of this data for that purpose is, how do you feel like you need to validate using CRISPR, and if so, how to sort of scale that process up?
Yeah. So I do feel that, especially if we want to make a biological point and really prove that the region is being regulated, we do two things usually, and I didn't, we did, there's a lot more validation in all those papers than I showed you. I just skimmed over one example for each, but I'd have to say that when we pick high confidence interactions, that is those that overlap with Hi-C and ATAC-seg, I'm fairly, we get, we haven't done hundreds, so I can't give you a number, but really a lot, most of them are validated. That's number one. When we pick things that, where we have an ATAC-seg enhancer promoter correlation and a Hi-C loop, those have been readily confirmed with CRISPR in the right cell type. Surprisingly so. And in fact, even what Heijung tried, most of hers validated even without the ATAC-seg. the majority. But again, I don't think we did enough to be 100%, to put a number on that. We also do reporter assays as well. So we ran some reporter assays just to show that the variant itself was actually changing expression, the SNP that we were looking at. But to kind of validate the high C, I think there are a lot of methods out there now that we've been toying with, but we're not expert in yet, which involve kind of multiplex CRISPR libraries to, and then doing single cell sequencing to recover the barcodes so that you can actually identify the cell type as well as the gene expression changes in that cell type. And so we've started to do some of those pooled CRISPR screens. I think if your goal is to test thousands rather than a few dozen, that's a way to go. And it also gives you the cell type resolution. There are others who are much more methodologically advanced than we are, who have been doing this kind of thing for a while. So we've started to do some of these pooled CRISPR screens. We're doing that when we have hundreds to, high hundreds to thousands. I think for a couple dozen, the CRISPR is easy enough you don't need to do anything fancy. I think just choosing the right cell type, primary cells being better than cell lines, because that's one of the points here, is that a lot of these things are stage specific. But does that make sense?
Yep. Okay. Well, I think given the time, maybe we'll pause the questions for now and I can hand it back over to Joe to wrap up. And thank you very much, Dan, for your excellent talk.
Wonderful. Thank you, Anthony. And big thanks again to the speakers, Shahram, John, and Dan. Thanks to the attendees for joining us. Quick promo, visit our Twitter account for latest or website for resources, links to pubs, testimonials, videos on our YouTube. Thank you, everybody, and have a great day.